Hey guys, so today we're going to dive into the basics of heredity for our AP biology class. Now this can be some people's favorite topic, it can be some people's least favorite topic, but hopefully we'll get the basics down pretty quickly and then we'll get into the good stuff very soon. So let's start with Mendel. So you may remember from your regular biology course that Gregor Mendel was whoops, that went away. Guess that linked to a quote. Oh well. Gregor Mendel was an Austrian monk who bred garden peas and brought an experimental and quantitative approach to inheritance. People had studied inheritance before, but it was really Mendel who brought this original scientific approach to it. And we later found out that DNA is the hereditary material in all cells. Mendel called it something different. He called it elements or elementes, which we now know as genes. And he studied what he called particulate inheritance to find out that these traits are carried by something within the organism. And later on, we found out that it was DNA. So this is the quote that I linked earlier. But my scientific studies have afforded me great gratification, and I am convinced that it will not be long before the whole world acknowledges the results of my work. And that was something that Gregor Mendel wrote back when he was doing his research. And yes, the entire world does acknowledge his work, and he really truly is known as the father of genetics. Now, we're going to look at some basic heredity and hereditary problems today, but there are some things that follow Mendel's rules, and there's some things that don't follow Mendel's rules. Before Mendel, people hypothesized that traits came from a blending of individuals, which we now know is not true in many cases. So we don't have purple flowers appearing because we have a mix between, pink and red or whatever other colors. We have traits that either can present dominance over other traits, or codominance or incomplete dominance, and we'll talk about those in a little bit. All right, so we talked about that. Really quick, back to reproduction. We already talked about mitosis and meiosis. Now, mitosis is our basis for asexual reproduction. Mitosis happens so many times a day within our own bodies, and it's going to produce two genetically identical daughter cells. Meiosis, on the other hand, is the basis for our sexual reproduction. So meiosis will produce our gametes with about half the normal amount of genetic material. These gametes are produced in our gonads, our sex organs, and it's going to have those particular traits, which we talked about, to increase variation within a species. I'm going to move my face down here. Okay. So these are the three things that we want to remember about meiosis and how we're going to get that variation. Of course, with mitosis, we can get variation through mutation, but that's the only way. Mutation can happen as well in meiosis, but these three mechanisms are how we're going to get variation consistently within the process of meiosis. So we have independent assortment. Independent assortment, we already talked about how these particular chromosomes are lining up either on the top or on the bottom. And to calculate the number of possible orientations of our chromosomes, we take 2 to the whatever number of our haploid is. So in us, our haploid number is 23, so we do 2 to the 23rd, and we have 8 million possible orientations of our 23 chromosomes. For an organism that has 10 chromosomes, where 2n equals 10, we would take the haploid number, which is 5, 2 to the 5th is 32 possible combinations. So that's how you calculate the amount of different possible combinations of those, or possible orientations of those chromosomes. We can also calculate the possibilities of random fertilization. Now this becomes even a bigger number. We do the same we use the haploid number n, 2 to the n times 2 to the n, and we're going to get the calculations for the possibilities for our random fertilization. And then finally, for crossing over, we're going to have functionally infinite combinations of information. So we can't really calculate a solid number for that because the number just gets out of control, so big. So that's why we say it's like infinite possibilities. We could calculate it, we'd be here a long time. Okay, so chromosomal basis of inheritance. The chromosomal basis of inheritance provides an understanding of the pattern of the passage of genes from parents to offspring. Now, of course, Mendel didn't know quite about chromosomes and how their structure was and everything, but he was able to talk about the transmission of this information from parent to offspring. So let's review really quick genotype versus phenotype. I remember genotype like thinking the actual genes. And these are the alleles that an organism has for a particular trait. So genotypes can be homozygous, homo meaning same, and then we have like big A, or little a, or they can be heterozygous, two different alleles, big A, little a. Now remember, our phenotype is going to be the trait that the organism shows. P for phenotype, P for physical. So think physical phenotype. So this will be something like blue eyes, or dark fur, or curly hair, or whatever phenotype you're going to talk about. Okay, so one of Mendel's laws, his first law actually, is the law of dominance, and we're going to talk about dominant versus recessive genes. So people were always confused about like how traits could disappear and then reappear in different generations. For dominant traits, we have an allele that is going to be expressed over a recessive allele. So when an allele for a recessive trait is present along with an allele for a dominant trait, the dominant one will be expressed. That is what our law of dominance says. In order for a recessive trait to be expressed, both alleles have to be present for the recessive trait. I think I'm missing a word here, but that's okay. So in order for a recessive trait to be expressed, both alleles have to be there. It has to be homozygous in order for us to see that recessive phenotype. Now, before we get into our laws of segregation, do keep in mind that there's other types. There's not just classic Mendelian dominant and recessive genes. There's also codominance, incomplete dominance, other things that we'll talk about, or genes that are linked together. But this is our main one. This is the one you probably remember from regular biology. Okay, so during meiosis, each gamete was going to receive one of the organism's two alleles. So each organism will produce an equal number of gametes with each allele. So, for example, if our genotype was big A, little a, we're going to produce gametes with little a, and we're going to produce gametes with big A, equal number. So, if we are looking at a regular old cross, this is a monohybrid cross, which, if we remember, we can do this easily with our Punnett square. We have our F1, which is our parent generation, our first generation. Excuse me, P is generally our parent generation. F1 is our first generation. And what we're doing is a cross between two heterozygotes. So we have an individual that has big T, little t, and we're crossing it with another individual who has big T, little t. And so this monohybrid, one hybrid, a hybrid is another word for a heterozygote, is going to be crossed with another hybrid, another heterozygote. And so what we do is we are separating each of these alleles individually and seeing what they would combine with the other individual during fertilization. So we fill out this Punnett square. I'm sure you remember how to do this if you went into a few practice ones. They're always fun to do. And then our possibilities for our F2, our second generation, are going to be big T, our homozygous dominant, two big T, little t, so two heterozygotes, two hybrids, and then one little t. And we're going to get this classic ratio for a monohybrid cross. We're also going to talk about test crosses. Now, this is also called a back cross, and it's a method to determine the genotype of an organism that shows only the dominant trait. And how we do this is we get, so we don't know, for example, like say the trait was, I don't know, dark fur. Say dark fur was the dominant trait. We have an individual with dark fur. We don't know if that individual is homozygous dominant or heterozygous. So we can cross it, actually physically do the cross, cause that organism to mate with a homozygous recessive individual, and then we can see from the offspring what the actual genotype of the original dominant parent was. So using this, we can basically determine what our mystery genotype is. So we could do a test cross. For this case, we have tall plants versus short plants, and if we don't know what the second letter is, the second allele, we can do a test cross by using the homozygous recessive and then seeing what the offspring is. If you get 50-50, 50 are tall, showing that dominant trait, and 50 are showing the recessive trait, then that means that this original parent individual was heterozygous. If we're getting 100% as expressing the dominant trait, so 100% tall, then we know that this original parent was homozygous dominant. Okay, so test crosses can be very useful. All right, so we already talked about how some of these traits are not on the same chromosome. So during meiosis, each gamete is going to receive one of the organism's two different alleles. And so we call this the segregation of alleles. And we're going to have an equal number of gametes, like I said, with the same chromosome. So if this looks complicated, it's because it kind of is. This is what a dihybrid cross looks like. So a dihybrid cross is a cross between two individuals that are hybrid for two different traits, or heterozygous for two different traits. And we're going to get 4 different types of gametes here. So if we have a parent that its genotype for 2 different traits is big R, little r, and big Y, these are 2 heterozygotes or 2 hybrids. And then we have the other parent with the same genotype. What we'll do is we'll put the possible combinations of all the different possible combinations of the gametes on the top, as well as the combinations of the gametes on the sides, and then we'll combine them all for the different types of traits. In a classic dihybrid cross, we are going to end up with the ratio 9 to 3 to 3 to 1. And we'll do a lot of practice with this in class, so if it seems complicated, it's really not that daunting. It's just a larger version of the Punnett square. And here's another version of it. We have these flies with either red eyes or brown color or black color, and we get the same ratio 9 to 3 to 3 to 1 when we do this dihybrid cross. Okay, so using this we can determine the backing up, the inheritance of unlinked genes, we also have linked genes, are independent events, similar to like multiple flips of a coin. So in order to calculate this, all we have to do is multiply. And remember, this is for unlinked genes only. We don't do this for linked genes. That's something different. But using this, we can determine the probability of a particular group or offspring being produced from a particular cross by determining the individual probabilities for each trait and then multiplying them together. So we can do an example here. I'm going to move my face out of the way. And then we'll do more practice in class. So this is a lot of text, but we can break it down. In pea plants, the gene for wrinkled seed pods, big R, is dominant to the gene for smooth pods, little r, and the gene for yellow seeds, big Y, is dominant to the gene for green seeds, little y. A heterozygous wrinkled yellow pea plant is crossed with a homozygous smooth green pea plant. What fraction of its offspring will be smooth and yellow? So let's break this down. This is what our cross looks like. We have a wrinkled, this is my wrinkles, a wrinkled yellow. So wrinkled, and remember we said it was heterozygous wrinkled, and yellow again. So we have our big R, little r, and then we have our yellow color here, and then it's crossed with, whoops, gave it away, crossed with a homozygous smooth green pea plant. So this is our green color, it kind of looks green, homozygous and smooth, so we have a little y, homozygous recessive for both genes. So we're going to cross these two. What we can actually do here, instead of, your first instinct may be to make this really big cross, which we don't want to do. We're actually going to look at the possibilities of each particular gamete, and then we can multiply those numbers together and get our final answer. So I kind of give it away here. But the possibility of us getting little r, from this particular cross is 1/2. Okay, so we can actually split this up and do the individual Punnett squares if you want. So the possibility of us getting little r from this particular cross of big R and little r is going to be 1/2 or 50%. So we're going to multiply 1/2 by the possibility of us getting a yellow pea plant. And so yellow, we look at the same thing. We're going to have 50% chance of us having an offspring that is yellow. So we multiply 1/2 by 1/2, which is going to get 1/4. So the answer here is there's a 25% chance that we will get a smooth yellow pea plant from this cross. Okay. This one looks scary, but it's actually not. Same idea. This is a longer list of genes, but what is the probability of getting an offspring with the genotype little a, big B, little c, big D, little d, and big E, big E? So we got a lot of different combinations, but what we're going to break down is the probability of us coming up with these two letters from each of these crosses. So what we'll just do is break it down by letter, and we'll go through this one in class as well. And then you'll come up with the probability for each particular different genotype within this long list of genes, and then we'll multiply all these together, and our actual probability of us getting this exact genotype from this cross is going to be 1/128. Okay, so that is our last practice problem for today. And I want to make sure that we do a lot of practice in class, so if you're a little confused, don't worry about it, and we'll do more genetics soon. All right, thanks guys.